started in just a moment. Just a minute longer to get settled. So if this is your first session, we have the conference Wi-Fi there. It's also on your schedule here. The Wi-Fi name is NU guest or NU wave guest, and then you have that. You can use that name and password that's posted there, as well as on your schedule here. All right, we'll go ahead and get started. Hello, everyone. Welcome to the Advanced R session of the May Institute. My name is Carey. I'm one of the organizers and instructors for this session, and I'm very happy to start off with a keynote by Vincent Carey. He is a professor and bioscience at Harvard Medical School and also Brigham and Women's Hospital, and he is also one of the co-founders and core members of Bioconductor, which is the R repository for bioinformatics-related software packages, which we'll be talking about a lot during the rest of the session. So I'm very excited to get started with our keynote from Vincent.
Thanks very much, Kylie. Pleasure to be here. And this talk is called Integrative Proteogenomics 2019. And I invite you to interrupt me when there are questions, issues of clarity, and so on. I've tried to spell out very clearly what's going to be done at each stage of this talk, because there's a lot of material. And the notion of advanced R programming, R software design, R deployment for scientific investigation, well, it's a big topic, and it may mean different things to different people. I'm going to try to start out with some scientific content that I think should be of interest to most people who are thinking about proteomics research. And so we'll start with this idea of proteomic annotation, or the annotation of genes using results of proteomic experiments. That's a pretty brief part of the talk. And then we'll get into how bioconductor concepts can be used to deal with data that are emerging in an NCI initiative to collect proteomic experiments for use in cancer biology and cancer therapeutics. And this is going to be a somewhat difficult part of the talk from the scientific perspective. There'll be a lot of things flying around. And again, if you haven't done clinical genetics, and I say something that doesn't make any sense, just stop me and we'll try to spell it out. Well, once we've gone through that scientific material, we'll start talking about advanced R. And one of the things that's coming up a lot in the domain of genome research, as the assays are getting finer and finer grain resolution, the single cell genomics domain, the reliability of experimental reports needs to somehow be systematized. There's been a lot of issues with reproducibility in large-scale science. And so this concept of FAIR, what does it mean? Findable, accessible, interoperable, and reusable. Somehow those four terms came out, and they are being used to describe sets of principles to help people who are doing work in data-intensive biology to have more reliable methods. So we'll talk about how Bioconductor helps to implement these principles. And some of the things I'm going to get at in the advanced R space are this idea of an integrative data structure for multi-omic data called multi-assay experiment. And the way we're going to use this is we're going to have some data that are big enough that we don't really want to have them local. It'll be the whole cancer genome atlas. It's going to be in the cloud. And then there's some local stuff because there's new material that never made it into the Cancer Genome Atlas or hasn't made it in there yet. We want to work with it. We want to combine it with the Cancer Genome Atlas. Then we'll talk about the idea of an app. So these apps are things that you just sort of point a browser at or you point your phone to it and computations happen. Things that are interesting can be done. We'll talk about how to program an app for some of this proteogenomic data to help us think about whether the assays are consistent with one another, when they differ, how they might differ. And finally, I'll talk about the metadata problem. It's very hard to work with experimental data if we don't know a lot about the experimental conditions. And that metadata is growing, getting more complicated. It's often not standard. And so we're going to talk about how to tame that problem. It's a lot of material. Supposedly, I can go until 3 o'clock. I hope I don't. You hope I don't. And we'll just try to go at a relaxed pace. Welcome your questions and comments. Now, yeah, what are we talking about here in terms of advanced R? So there's a concept in R called S4. It's an object-oriented class system. So if you're a software developer, you've undoubtedly thought about the difference between object-oriented programming and other species of programming. If you're not, well, then you're lucky because we're just going to take care of that for you. Okay? And I'll describe to you how some of these data structures can help make your life easier with respect to genomic computation. Then there's this notion of edge computing. Cloud computing used to be the big thing. Now it's edge computing. Edge computing is where you combine local things with cloud things in a way that is very nice and seamless. And it's often referred to in connection with the Internet of Things, but you can also make use of it in data contexts. And then, yeah, advanced R. How many folks have used Shiny or think about Shiny? Oh, a good number of you. Well, that's good. So browser-driven. You know, the browser is something everybody knows how to interact with. They may not know about R. They may not use RStudio and so forth. So we can get sort of a common denominator of interfaces by working with a browser. And Shiny helps us to take our computations and our data and work with them in a browser context. We'll talk about some examples of that. And then there's the idea of interactive graphics. So I've gotten to the point where I'm sick of printouts. I mean, I've been sick of them for many years. Tired of printouts. They hand you a stack of printouts, you flip through them, you find something that looks interesting. It's a pain in the neck, and it's a waste, and oftentimes you didn't get what you wanted, and it'd be better to be able to compute immediately to find the thing that you're actually looking for. And so this idea of Shiny apps and then interactive graphics, where you see something in a data plot, you say, well, what's going on with that point there? Well, do you have to go back in and do some programming to find out what's going on, or can you move your cursor over that and get some metadata about what's there? So we'll talk about how to do that. How many Plotly users are here? A couple. All right, good. And then, forget it. I don't want to work with my laptop, or I don't want to make people buy computers and so on. We just want to have resources globally accessible. You can call them in the cloud. They're served by somebody, whatever they are. I want to make my data and my analyses available to people, both in terms of push, here's what I'm telling you, and pull, these are things that you can do with the resource that I'm giving you to learn things that I didn't think of. Okay, and that's where these open apps and notebooks are going to come in. So it's a lot of material, and it's going to come to you. I'm saying it again, 3 phases. First, the scientific context, thinking about peptide distributions in human tissues, and then DNA variants. Then we'll talk about packaging data in R code to do this in a fair way. And finally, we'll talk about how to get these things into positions where you don't need a special environment and data structure and so on to interact with data. And so there are some acronyms. I want to make sure we get through them and I don't confuse you. TCGA is the Cancer Genome Atlas. It's funded by the NCI. It's a number of years old. 10,000 plus tumors have been assayed in many different ways, proteomics being one of them. And the way in which the proteomics was done for many of the samples is called the reverse phase proteomic assay, a proteomic array, reverse phase. So you have to know in advance which peptides you're interested in quantifying. You put them on an array, and then you do some assays, and you get some numbers. And there's a lot of RPPA data in TCGA. We'll talk about it from a different quantification perspective, iTrack. I don't know much about iTrack, but maybe you guys who are studying proteomics do. How many people use iTrack quantification? A fraction of you. So this is something which, some of you are going to have to help us understand it better, because I don't have a rich understanding of it. It's not absolutely critical that we understand it completely, but what we're going to say is we have some iTrack ratios that have been collected on a number of tumors. We're going to assemble them and try to interpret them in certain ways. So RPPA, I mentioned to you, RNA-seq is looking at messenger RNA abundances in tissues. We're going to only talk about bulk RNA-seq here. Diagnostic subtypes. Breast cancer is a whole collection of heterogeneous diseases, and subtypes of breast cancer have been identified. We'll talk about terms that are types of breast cancer that are identified through transcriptomic studies, the so-called PAM50 classification. It's not crucial that we understand them completely, but this is something that we're going to want to work with this data alongside of. And finally here, yes, NCBI, the Bioinformatics Institute or Center in the NIH, has something called the Sequence Read Archive. And we're going to harvest the Sequence Read Archive for metadata and data. And those are a couple of acronyms that I want to make sure are not confusing to folks. In the computational side, we'll be talking about S4. And then Google has a very interesting SQL-like database system called BigQuery. We're going to be illustrating how that can be used from the R perspective with very little concern with BigQuery itself. And then ggplot2 and Plotly. These are R packages that can be used to help build dynamic visualizations. And then when we get into the really big data, we'll talk about HDF5 scalable data service. So the HDF data are put in object stores in the cloud, and then we can have many, many data points. And finally, Google Collaboratory is something which we will illustrate for the use of notebooks. Okay. So article number 1 here is this thing from 2016, the GenCode reference gene annotation using a proteogenomics workflow. And what was helpful to me in this context was, first of all, the idea of the quantity of information, right? So from 30 tissues in 85 experiments, 25 million spectra have to be processed. Add them together, because we're getting them from 3 different labs, and go through this workflow, and ultimately you can get some information on 16,000 genes. That's what GenCode is, really shooting for, trying to have very good annotation of human genes. And you're starting out with 17 million peptide spectrum match data. Okay, I'm sorry that that's so down low there. Can everybody see that? Should I? I'll do it like this so you have a better chance of seeing it at some scale. Whoops.
There is a link to these slides on the Pinterest.
Oh, yeah.
Under resources.
All right. Yes, you can get the slides and make comments on them. All right, so that's the workflow. And all of you are going to be engaged with either those workflows or just the products of the workflows. We'll just think about the products of them. So again, we're talking about the scope of information that has to be managed in order to work with this stuff. This is a histogram giving how many distinct peptides are found in different human organs. So it goes up to about 160,000 per testis. And a lot of these tissue types of great interest, B cells, for example, have close to 100,000. T cells, just the CD8 type. So we want to understand the biological function of these things. One place to start is the peptide distribution. And these are the numbers of peptides that have been found. Then the number that are unique, found only in a given organ, is about an order of magnitude smaller. So if you're going to think about things that are specific to the organ, you're going to be fishing around for these guys in the context of 10 times as many peptides. And the reliability of the work that we do is not absolute, right, in this space. So there's a lot of information that you're going to be sifting through. And this is a very basic problem, right? Set up the workflow, use proteogenomics to help catalog our genes. Eventually, we get a new catalog. We have to reannotate everything. This is the business you're in. And this is just in normal samples. So now I want to switch from this problem to cancer, right? We talked about normal. Cancer, if you have any experience with it, the disturbance of the genome can be extraordinarily complicated. And the question is, can we learn about how to deal with that from a medical point of view using proteomics? So this is a paper from 2016. And we're going to look at the workflow that they've got here. So you have 105 breast cancer samples, which are processed through this workflow. And finally, we get some quantification of peptides using iTrack. And we go through all of this, and then we do data analysis in spectrum mill, which allows us to get these iTrack relative peptide abundances at the gene level. We're going to take this information and boil it down to peptide abundance at the gene level. That's boiling down a lot, right? So even that is challenging to work with. And I want to make sure everybody feels comfortable with that at the end of this talk. So here's the abstract. There's a lot of interesting concepts here. You have 105 genomically annotated breast cancers. 77 of them provided high-quality data. The others were omitted. I don't know whether that's a typical ratio, but that's a lot to lose when you've done an expensive assay. Okay? And what were the criteria? What if you kept one of them and you had 78? Would the story change a lot? If you used different statistical methods, could you have used 85? These are the kinds of things I think we want to be able to answer, certainly in our own experimental conditions. And when you say, well, we want this to be fair, findable, accessible, interoperable, reusable, you really want to know how we came up with that 77. Now, consequences of chromosomal loss, like the 5q deletion, what can we do in terms of the impact on peptide distributions or protein distributions when we have a piece of a chromosome deleted? How can we interpret that in terms of protein effects? And one of the ways they did this is to use this thing developed largely at Harvard called the Library of Integrated Network Cell-Based Cellular Signatures to try to figure out what's the effect of losing some crucial genes that are down on 5q. The conclusion, proteogenomic analysis of breast cancer elucidates the functional consequences of somatic mutations, candidate nominations for driver genes within large deletions and amplified regions, and identifies therapeutic targets. So those are great results based on a relatively small sample size, but very intensive proteomic assay. So those are the highlights of the abstract. And I'm not going to get into one of the dimensions of this talk, which is that we don't just look at the proteomics, but we look at phosphoproteomics. So that paper's available to you. You can start getting a handle on that. And I'm not going to get into that. So here's one of the nice figures in the paper. We're not just talking about proteomics. We're talking about proteomics in the context of DNA modification related to a disease. How do we measure these modifications? Well, there are many different ways. You can say, well, there's variation in DNA. And it could be germline variation, which is not necessarily interesting, although the combination of germline variation and tumor variation may be very interesting. And then what's really critical in thinking about these is called somatic variants. These are changes to DNA that are local to the tumor tissue. And here are the relative abundances of these events in the 105 samples or 77 samples that are ultimately going to be analyzed. Variation in DNA, also observed in RNA. Sometimes variants are observed in RNA that they can't track to a DNA change. And then the critical point is that we can also use proteomics to see that there are variants in the peptide distributions that one would want to interpret in terms of biological effects. So you're boiling down from 35,000 DNA variants, maybe 70,000, to 3,100 or 7,000, what have you, depending on how you want to look at the proteomic changes related to DNA variation. So that's one type of variation of interest. Now another type is where there's some differential editing going on between the tumor and the normal tissue so that you have new splice junctions in the data that are found in the tumors. And these, of course, have effects on the protein and peptide distributions. And these are the numbers of events that you're going to be thinking about and trying to interpret. Okay? And here are some examples. So again, boiling down to the gene level, here's an important oncogene TP53. And what we're listing here is how many of the samples had a germline variant in TP53, how many had a somatic variant. Very small numbers. And what is the actual nature of the change? So that's the way they give you access to the variants. And now we can look at the overall effects of copy number changes. In this case, this is where genes have been amplified or deleted, and possible effects on proteins local to the change or remote from the change. These are called cis effects, and these are called trans effects, where they're far from the DNA event. And so you can use RNA sequencing to identify effects of these changes, or you can use protein quantification to identify these. These are often very consistent. And then using this experimental system called the Library of Integrated Network-Based Cellular Signatures, you can say, well, when we've knocked out the region that holds set N3, we can find effects on proteins that are far away from it, trans effects. And the hope is that some of these will help us figure out what's going on in these tumors and how we can treat them. Now, this is the data resource provided with the paper. There are 19 supplemental tables. Some of them are numbers of megabytes in size, Excel tables. And it's not obvious how one would go about analyzing these, so there has to be some transformation of the data. And that's what we're going to work on next. So to summarize where we've gotten to now, we have this TCGA. It's a potent resource for many different types of cancer. We're going to focus on breast cancer. There's RNA-seq done on 1,000 tumors, and there are some tumor normal pairs. There's reverse phase proteomics on 873 tumors, only 259 features, and the iTrack quantifications involve 12,000 features on 77 tumors. And this is where we have boiled the data down, or we've used their reduction of the data to gene level. And the reason that I'm getting involved in this is to pose to you, and to hear more questions from you, is this a good thing to do from a biological perspective? Can the effects of these variants be more sharply characterized if you're doing the analysis as opposed to the authors? And can these methods be adapted to other tumors and diseases? this is why we would like to have sort of a mechanism for looking at the research artifact of a paper like this and abstracting out the components that would be common across different tumors, let's say, and making sure that we don't have to reinvent the wheel when we start working on prostate cancer or some other cancer, but we can take these tools and deploy them readily, substituting in the TCGA data plus some proteome data from some other tumor initiative. All right, so that's the end of phase one. That's the science, the biological science. Comments, questions? Okay, so we're going to transition to R. Now that wasn't bad. We're going to transition to FAIR principles and the way Bioconductor helps you implement them. Okay, so Findable. What's Findable? Well, I can do a Google search and then I find what I'm looking for. Accessible. I can download it. No. I mean, that's old, right? Interoperable. Now that's interesting. What does that mean for a research artifact to be interoperable? Well, it doesn't require any special coding, let's say, to use it in some interesting way. You know, the way the data are set up, let's say it's an Excel spreadsheet. Well, is that interoperable? Well, it turns out that, yes, there's an API that you can use to throw information at the thing that is running Excel, and it will give you back information in a way that it doesn't require you to be running Excel. We're using the statistical methods inside Excel. Okay? So that's an interesting idea. And how do you carry it out? Well, I guess, you know, in many ways, the concept of an API or an open API is the thing that's going to drive folks in the direction of saying, well, I've got some standards that I need to meet here, and I'm going to make it an interoperable data resource. And that means that if you host it in a certain way, then certain queries can be put in the form of URLs, and things will happen, and you'll get interesting data. And that's something probably worth learning about, but it's still pretty difficult to negotiate. There are tools in R that will help you do that sort of thing, but I'm not going to get into that. I'm going to assume that things are made interoperable enough so that there are very simple things I can do in R using packages that already exist that allow me to get things and do some data analysis. And then reusable, I don't really know how to distinguish that from some other features, but that's how it looks. And if you want to read the paper about this, there's a link for it. Now, I have no shame in saying that I think we've been doing this for 20 years. We started out when microarrays were a big deal, and they were big, and they were unwieldy, and they were poorly annotated, and so on and so forth. And Bioconductor came about in order to help statisticians understand DNA microarrays and to help people who are using DNA microarrays do the right type of statistics. We started that in the year 2000. And so we had our repository with all kinds of wonderful continuous integration and vignettes and so on and so forth a good 15 years ago to solve the findability and accessibility problems, both for software and for data annotation. And by now we're using Git. How many GitHub users out here? Good number of you. Okay, that's good. So Git, digital object identifiers, R packages, and so on and so forth. We've been doing that for a long time, and it really does solve those problems. Interoperability through shared infrastructure, the things I'm going to tell you about, people put their new experimental data in these structures, and other people can go and start working with them without having to parse them and find out how things coincide and so forth. And then there's continuous integration of the whole ecosystem. So there are 1,500, 1,700 packages in Bioconductor, and they all have to be consistent with one another in some sense when they do interact, and that consistency is tested. Finally, reusability, well, that's mainly through openness, right? If I do something, I make it open to you, and you can do the same thing, or you can change it a little bit, and then you can do your thing. And GitHub plays a role. This is the BRCA Proteo GitHub repository, open to the world, where I have done these things. And if you look at it now, you'll see that there are changes in the last couple of hours as I finished up the talk. And this is something you can use. So it's structured as an R package. So you'll see that there's a description file here. And this is a bit of advanced R. You're not thinking about programming R. You're not thinking about running R and typing things. You're thinking about making a piece of software that will work in R in a way that it can be shared easily, because anybody who finds it can install it because it satisfies certain conditions. It has a description file, and that's about it. If there was some R code in there, then you'd need a namespace file. But if it has data, then it will deal with that data and put it in the right place. If it has a vignette, it can go and compile the vignette and tell you things about it. So this folder structure and certain constraints on what these files need to look like are simply articulated, easy to comply with, and that gives you the ability to write software that can be run by anybody who wants to use R. Okay. I'm not going to get into package construction. I think that'll probably be entertained somewhere else in the course. There's lots of material to help you with this, and I'm going to take this for granted. An R package has all kinds of folders, and then we can use a documentation procedure so that within each one of my source code files, I use a certain syntax, and it will generate manual pages that describe how each function can be used. And if I have some data that I think you may want to use without going through the process of reading it in and annotating it and so forth, I can put those data elements inside the data folder, and you can just load them up and they'll be ready for you to go. And then Markdown, R Markdown, can be used to set up a vignette, and I will talk to you about those in some detail. Once I've made my GitHub globally accessible, then this is a command. There are other commands, but this will install it. And in fact, if you use this, it will do a pretty good job of tracking down all the dependencies that have to be resolved in order for the package to work. So we've worked hard on putting this type of infrastructure together. And in fact, in the year 2000, a lot of this stuff didn't exist. So Bioconductor was founded by Robert Gentleman, who is one of the co-creators of R. And he was able to help us implement a lot of these things so that we are where we are today. He's over at 23andMe now, but we started this out when he was over at the Farber. So this is what a vignette looks like. So once I've compiled the vignette using render on the R Markdown file, I can mark it up so that I have a nice hyperlinked table of contents. And this proteogenomics data archive has an introduction, a multi-assay experiment definition, and then a sanity check. So let's get into some of that. This is what the data looks like. So I've stored here a ProteoMAE, proteomics multi-assay experiment, and it has 3 major experiments in it. It has an experiment called iTrack, which has 12,000 features on 80 samples. RPPA, which is the reverse phase proteomic array, 259 features on 873 samples. And RNA-seq is transcriptomic study of 1,097 samples with 20,000 features, gene level. Okay? They're all together in one entity, and the syntax of working with this is partly specified here. So this is what's called the show method for a multi-assay experiment, and it gives you a bunch of hints about things you can do with this object. So for example, you can run experiments on it, and you'll get the list of these summarized experiments. Now I haven't talked about what a summarized experiment is. It's a matrix-like thing. So it has rows and columns. And the rows model features of your assay, and the columns model samples. That's good. But that would be a matrix, let's say, that has the quantifications. You also need some kind of entity that tells you, well, what are the features? So it can have something called row data. And then what are the samples? That's something called col data. And both of those can be data frame-like things. Okay? And experiments will retrieve those summarized experiments as you like. The col data is the primary data that may describe all of the samples that are in this collection of experiments. And the sample map is a technical thing that allows there to be relationships among samples that are in the different assays, you can see there aren't the same number, so there has to be some way of collating the ones that are exactly the same tissue, or tissue donor, let's say, that has been assayed in the 3 different domains. More interesting is that we can actually use bracket notation. So if you're an R programmer, you know you can take a matrix, you use a bracket. The first index refers to rows, the second index refers to columns, and you can get sub-matrices using that syntax. You can get sub-multi-assay experiments using the same sort of syntax. And then there are other nice things that help you restructure the data if you need to do that. So if you're doing multi-omics, multi-assay experiment is not a bad place to go. And TCGA is intrinsically multi-omic, so we can generate multi-assay experiments for any of the tumors in TCGA with any of the assays that you're interested in putting together, and you'll have access to all of this infrastructure to manipulate the data. Now this is something called an upset diagram. It's like a Venn diagram, but it allows more complications, easier to parse. So this is telling me how many samples contribute RNA-seq, how many RPPA, and how many iTrack. And then these things tell me how many are coincident. So only 76 samples were assayed by all 3 methods. 795 by 2 of those methods. Those are the ones that are in TCGA. And there's only 1 that has some RNA-seq and some iTrack. So this is a high-level operation. You give me a multi-assay experiment and you want to know how the assays lay out among the different samples that are in there, you can just run a function called upset samples and it will show you that. Now one of the problems with the data as they are conveyed by TCGA is that they use different annotations. So genes here are annotated according to the entree vocabulary, which is just to use integers to enumerate the genes. We need to remap them, and we can use Bioconductor to do that. So if I want to map from entree ID to symbol, knowing that that is a somewhat fraught thing to do, I can use this code. Okay? And then I just substitute symbols for the original identifiers, and now I have symbols instead of entree IDs. And so this is what we start out with before changing all the different annotations so that they are consistent with one another. And this is what we end up with when we both do that and then intersect the samples and the features to those that are common across the 3 assays. Okay, and all of the code that does this work is explicit in the vignette. So you could go line by line through the vignette to generate this object from the original one. Now, things are going to get a little more interesting, right? This is the kind of thing I suspect many of you have had to go through. Filtering data, making 2 data sets that come from different places consistent with one another so that you can learn about comparisons between the 2 different experiments. And we've tried to make this high level so that if these guys satisfy certain conditions, then that intersection process, that harmonization process can be done using high level, reliable code. But, I mean, that's advanced R, but it's there for you. You can understand that without any difficulty, read the documentation, so forth. Now I'm going to push into the edge computing concept. Because this data structure here says, well, you know, I'm working with this one here in particular. I'm working here with 1,000 columns and 20,000 rows. That's a lot of data, and on a small computer, that might be rather taxing. Okay? We don't really want to do that, and we want to be able to handle very large numbers of rows. And we can do that because the data are actually in the cloud. So that's where this edge computing thing comes about. We did all kinds of operations, but we didn't actually change the data. We didn't do anything to the data on RNA-seq or RPPA. Okay? All we did was manipulate the metadata that's sitting in our R session. What's going to happen is only when we really need the quantifications that are in the cloud, will R actually go and fetch them. Okay? And there's a lot going on under the hood there. There's a lot of skepticism that should be in place. You should require tests and so forth of these operations. But this is where we're going to go. The RNA-seq and the RPPA data are in the Google BigQuery for all TCGA tumors. Now that's a nice thing, because that means if I've solved some problem for one tumor, I just have to make a simple change. I change the code, the so-called acronym, and I get all the data for other tumors. Okay? But the things that are doing this are infrastructure packages called delayed array and RESTful SE, and that's what allows us to work at the edge. The metadata is local, the real data is remote. Not only that, but the proteogenomic data is not in BigQuery. It's not really part of TCGA. So that's local too. So here's what things look like when I start wanting to see the actual numbers. Now, in order for this to work, you have to have a Google Cloud Platform billing code. Okay? And I'm not going to go into that. Maybe later, if people want to talk about how that can be accomplished, we can do that. But once you've got that, you can go through this little dance. Here we are. We're looking at the BR proteome MAE that I've showed you before, and we want to query it using the assay function. So the assay function is going to go in and get some quantifications. And if you don't tell it which ones to get, it will try to get them all. But because of the delayed matrix interface, the assumption is that you would never want to get them all. You'd have to explicitly ask for them all. And so what it's going to do is set up a query that just gives you the corners of the big matrix that you're looking for. But in order for that query to resolve, you have to tell BigQuery that you're going to pay for this query to be resolved, and who you are, and how you're paying for it. And that all occurs through this authentication process, which is actually pretty simple. As I say, really all it needs is a valid billing code. And these activities cost fractions of a penny, very, very small fractions. Okay? But it is possible to generate larger bills if you do certain things in this space. So this is just illustrating how our infrastructure in the HTTR package and Google BigQuery can interoperate with your browser or other tools to authenticate you so that you can actually work with BigQuery and get some data out. Once you've done this once in a session, you can do it again. So here I'm using a somewhat more civilized approach to get the data. I'm asking for the RPPA data from this object, and I find that I'm getting a delayed matrix back, and I'm just getting the corners because I didn't give anything more specific. I can see that I have nice feature names here. They are symbols. Over here, this is at the early data. This is not the data that have been harmonized, so the symbols that are the rows of the data are actually the entree identifiers. But inside TCGA, for the RPPA, they actually use the gene symbol, so nothing had to be done in that case. Now, as I mentioned, the iTrack data are not in the cloud. So there's no delayed. See how this thing is telling me, oh, this is a delayed matrix. That's not the case here. And so I'd better go and limit the number of rows and columns I'm asking for here. I do so, and I get back these quantifications. And you can see that another nomenclature was used here. These are the RefSeq IDs, and those have to be remapped in order to use the symbols. And that's written up in the vignette. Okay? All right, we're almost done. Now we need to know, what are these samples? What are they like? And that's where the call data method comes in. And in this case, I'm asking for the call data of the third experiment. And I'm going to get just certain rows and columns from that. And the important thing here is that it can tell me whether these individuals are alive or dead, how old they are, and so on and so forth. So that information is in there. Okay? So there's a summary. Row names are like row names on a matrix or a data frame. Call data works on the multi-assay experiment. reflecting the basic summarized experiment idioms. And there's a paper that we published describing this whole multi-assay experiment idea. So let's go back to the vignette and start getting into the numbers. One of the basic questions is, if you give me a gene, can I check to see how tightly correlated the different quantifications of gene expression are? Right? So the RNA-seq data have values. Yeah. Log of 1 plus the quantifications that are given in TCGA. The RPPA data are taken as they are, and the iTrack data are taken as they are as well. And what we've done here is we've picked a gene. It's BICL2. And this object here, Brint, is that object that we filtered so that there's only common features and common samples. 76 samples, 60 some odd features. And the features are different genes. So here I've said, give me the BICL2 features in this multi-assay experiment, take the experiment's structure, apply assay to them to get the numerical quantifications out, and use a summarizing apply, because this is going to be a list, in order to generate a matrix. And when I run pairs on that matrix, the pairs function in R will do this pairwise scatter plots of all the columns of the matrix that you give to it. This is what I get. And it's actually a pretty satisfying result. There's no question that there's positive correlation between these. There's also variability, and understanding variability or the extremes of variation is a type of work that we'd like to do. Okay? So my claim is that this is a very concise approach to getting information on a gene that helps us understand, do a sanity check, I would say, on both the underlying data and all the computations that we've done. Because there are a lot of them and they're very complicated. And I wouldn't be surprised if one of them was wrong, and then I shouldn't see this. This doesn't guarantee that it's right, but it's not bad. Well, that's BICL2. How about a different gene? How about an important gene in breast cancer called BRCA2? The story is not so good. Is the code wrong? Is the data wrong? Is the biology so different that maybe we shouldn't expect high correlation among these assays? I don't have an answer for that. The data are there. I don't think this is mentioned in the paper. I don't know that it's biologically important or not. Yes?
Quick question. I'm trying to understand when you combine all three data sets, you had like isoforms.
Yeah.
How did you combine them?
Yeah, that's a superb question. And the answer is, there's been a lot of reduction upstream. And let's just take a look at this. Because I wanted in this talk to go to the isoform level and have a piece of one of the apps that we're going to do, present the data not in this boiled down form, but in the more, because it could be that this is a lousy isoform of BRCA2. And there are others that are well correlated with the other assays. I agree with that. And I'm not going to jump around too much. It's my feeling that it was boiled down to a single BRCA2 representation in the matrix that I took from their Excel spreadsheets. But I'm not 100% sure. So we should table that one and build up the vignette if we find that there's another way out. Okay? But it's an excellent point, and I'm glad you brought it up. We might have time to dig through the Excel spreadsheets and see, but let's keep going and maybe come back to it. Okay. So, one of the issues in advanced R is that I want us to get away from static printouts, static graphics. And so that's where Shiny comes in. So inside this package that I've described to you, we have an example. We have all the code that will generate something that will build these plots for you for any gene that you happen to pick among the 60 some odd that are actually common across these. All right? Now, an advanced R task would be to make this thing a little more flexible so that maybe it was missing on one of these assays, but the other two were there, and why don't I just plot in that case, right? So then you'd be working with the data to somewhat more primitive level. You should be able to do that. So there's a question of, what do I do to operate on the data to make my computations easy, or what do I do to make the computations more interesting when I leave the data in a more primitive format? There are trade-offs. Okay, in this case, I said, I'm going to make the data simple, and then I can do this. I can build this app. I think it's easy enough to understand how this works. It's a list of genes, and when you pick a different gene, you get a different plot. Now, another embellishment would be to put some statistics here, and this pairs function is not particularly good at enabling people to annotate the data. So here's something that's a little better. What is this? This is ggplot. And now we've gone a lot further, but we also see some complications. So I'm not a ggplot expert. But I know that I want something which is putting the data, juxtaposing the data in some way. And I need to annotate it. So I need to tell you what assay is being plotted against what assay. And I want to tell you even more. So I have this information on the individual tumors. Some of them have a certain transcriptomic signature associated with them. And these are important because they lead to different treatment decisions for people, lead to different prognoses for people. Okay? So it's nice to have that information there. And there's not a ton of data. But looking at this, you don't see a lot of structure. I guess you would say, well, the basal-like seem to have lower levels of whatever gene this is, BICL2, than the other ones, potentially. Right? We want to foster this sort of thing. There may be other variables that you'd like to put in there. And this app concept gives you a lot more power to work with the data interactively than generating printout after printout. Of course, you can sift through many printouts, but it gets very difficult. So this is a function that's in the package. It's called plot3. But we want to go even a little further. So now if we use Plotly in conjunction with ggplot, we get a browser-based graphic where I can now hover over data points and get information. In this case, it's not really anything that you couldn't figure out already, but you can add additional metadata that will get printed when you move over these plots. And it comes for free. If you know how to make a ggplot, you can run ggplotly. It's not really free because you have to put a little more information about what types of things you'd like to have on the metadata, but this is a useful functionality in my opinion. All right. Yeah.
So this interactive plot, is that some ggplot code that you wrote, or is this something that is aware that you pass a multi-assay experiment, synchronized experiment, and knows where to find that?
Super. Absolutely super question, and the answer is the worst possible answer. Okay? Everything is coded here so that this talk looks good. All right? So let us go to the web, to the GitHub. Okay? GitHub. Oops. All right, where do we want to go? GitHub.com, VJCITN, BRCA Proteo. Okay? Let the code speak for itself. All right? So I'll blow it up so you can see it. But this is plot 3 here. Okay? All right. So here we go. Plot 3. Now, for those of you who are new to this business, this is what Roxygen markup looks like. Okay? Here I am writing down the parameters to this function written this morning. So let's look at it. It's not, do not do this, okay? There's bad stuff in here. Not in the sense that it's unreliable, but it's just bad form. This tx equals none. what's the idea? I wanted to have an option that will either take z-scores and plot the z-scores. So you simply take the data, subtract off the mean, divide by the standard deviation. You get some data that are relatively well behaved. And because the RPPA and the RNA-seq and the iTrack can be on different scales, you really need to do some kind of thing to make them. And this is a very innocent thing to do, to put them all in the same scale. And that's the default is that I don't do that. But if I put anything other than none, then it will take z-scores. Okay? Bad. But that's that. Now what's going on here is we're assuming in this code that Brint exists. And Brint is that multi-assay experiment that was boiled down from the original data. Okay? So there's no cloud involved. There's no filtering or whatever. And this is terrible. I mean, we're building up a data frame based on, you know, some S-apply and so on and so forth. So you can do this. There's a lot of stuff that's taken for granted. For example, the PAM50, that's that basal, the tumor type and so on. That's all hard-coded here. We don't want to do that. You know, you'd want to pick the variable from some list of variables that you'd like to color the points with. Okay? So you see what I mean when I say it's the worst possible answer. It'd be nice to have some mechanism for saying, well, I've got a multi-assay experiment. I want to do pairwise plots. Here's the feature. And I would suggest that it's not much more than this to do even that. But I'm not going to go all the way back and do it that way. I had to do something that was more seat of the pants because I had very little time. Thinking of the abstractions that you want to have access to in the multi-assay experiment. Let's say you take that upset plot that showed you how many samples have different, have common assay contributions and how many are different. That's an easy one, right? And now we're saying, well, maybe we want pairwise plots, or maybe we want some three-way plots that are, you know, manipulable in other formats. That's what I would really like to motivate here. You know, you have some multi-assay data. There are certain things you want to be able to do, and how can we identify them as high-level operations on these multi-assay experiments? All right. I'm almost done. What are we doing? Yeah. Present. Okay. So I just want folks to appreciate that it is rather straightforward to give your analyst and even yourself a lot of power to interact with the data that have very complicated provenance and possibly complex data structure and produce something that is dynamic and reusable and informative about the biology that you're interested in. So exercises. Modify PG consist to use plot 3 because it's a better way of visualizing. Add functionality to plot 3 to allow flexible choice of the coloring variable. Add functionality to record the IDs of points that you might have selected and claimed were outliers. And maybe other ideas. So these are ideas related to the abstraction, things that you'd like to be able to do for any multi-assay experiment. And that's good. That's the end of phase 2. Okay? So it's been an hour. Do you need to take a break? I've got a little more to talk about. Yes?
So I had in my project, so I had transcriptome and I had proteome and I have a couple of other things. And every time I wanted to do a scatter plot, or whatever thing, so I would annotate independently the proteome data, annotate independently the transcriptome, so I would have some name in common. And then for all this
Yeah, this is a great question, and I don't want to oversell this. You're talking about this piece here, right? Yeah. So this is why I put the code there. And, you know, Laurent asked the right question. You know, you said something nice about Brint, and there is something nice about it, but it's throwing away a lot of information that's in the original thing. It's doing it in a way that is very easy to do, and if you need to do something else, you're going to have to write something a little more complicated than that, okay? Now, what I think you were getting at is that if I were to do this type of work, how do I specify the information that will come out when I hover over this, right? And I don't know if there's a really clean way to do it. There's a bad way to do it in the sense that the ggplot guys don't want you to use this parameter called text. But if you do use it, then the plotly guys will be happy and they will put it there. So by setting up the text from data that's consistent with what you're plotting, you can have any annotation here you want. In fact, you could have a URL that a person could then click on to get some more information. But, you know, that stuff is advanced R that hasn't been done by enough people who are interested in it to make it something that's high level and, you know, easily reusable. Okay? So, wonderful questions. If you run into a brick wall trying to use these things in order to make your life easier, file an issue with the GitHub or send me an email or whatever, and I can't guarantee that there's going to be an easy answer. A lot of things are simply hard until we get more standardized tooling. Okay? Good question. All right, phase 3 is where we go into metadata and apps and other things that I think are important, that we get control over this and that we have people working with our stuff. There's this concept of mileage that I really like in software, right? If you write a piece of software, it has to be used a lot in order for you to find all the bugs in it, because they're definitely there. And that's why, you know, I believe this dictum, release early, release often. It's embarrassing. I put code like that out there, and I'm sorry, you know. I have limited time. I am a limited intellect, and, you know, you find problems, you can flame me, you can do whatever you want. I just want these things to work better, and I want them to be usable to people who can use them. And it's funny how many professionals I work with who will not accept that idea. Things have to be really perfect before they'll expose them. They don't expose them, you wind up working with imperfect things. That's life, you know. I read a book a long time ago called The Psychology of Computer Programming. It's by a guy named Gerald Weinberg. And he uses this term egoless. You know, he says in order for software development to be good, you have to work with people who are egoless. And there are these references to Zen, Buddhism, and so on and so forth. You wouldn't think, and I don't think that book is in print anymore, but these are sorts of things which I think are really quite relevant to how we're going to have to do this, because the data is mounting. We need more software, but it needs to be good. And in order for it to get good, you need mileage, you need bug reports, you need fixes, you need continuous integration, all the things that Bioconductor has been doing for a long time. And it's gradually getting to be the way of doing these things. It's not perfect, and we need improvements. So let me just finish up. This is an app called Cancer 43K. 43,000 cancer transcriptomes which are not in TCGA, but are in the sequence read archive. I want you to be able to work with them. How can you do it? Well, I wrote an app, and it's on shinyapps.io, and if you fire it up, you fire up shinyapps.io cancer CA43K, you'll see this. Maybe I'll do it in a couple of minutes to show you what's going on. Now, what's important about this is this idea of searching studies. You could Google on BRAF, I think that's how they say it, BRAF. You could Google on it, and you'd get all kinds of wonderful things. You can go to the SRA, and you can search for BRAF, and you'll get all kinds of wonderful things. What I want to get is a summarized experiment for studies that looked at BRAF. And I have the option of any of these 43,000 may have looked at BRAF. That's a pretty reasonable request. It's an important oncogene. People are studying it all over the place. I just want those studies. How do I get them? There's no answer. You can go to the SRA and you can start finding things and pulling them down. What I wanted to do is just find them and get them in my R session. Okay? That's the idea. Well, how do you find out which ones are actually studying it? Maybe they mentioned BRAF in the title of the study. Well, here's one where they did. But here are a couple where they did not. This is a RAF kinase, so maybe that has something to do with BRAF. Oh yeah, they mentioned it here. But this one did not. Okay. So the titles are okay, the abstracts are okay, but maybe it's not even in the title or the abstract. Because those titles and abstracts may be in very general terms. Well, we need to go into the metadata. And we go in and we search every single one of these tokens, and we see if we find BRAF, then they're in my list. Okay? That's what this app is trying to do. How do you do that? Well, you have to process the metadata. I'll tell you a little bit about that. Okay? The other thing is, I want to be able to search in a nice way. You know, I start typing something, and I want a suggestion of what's relevant to this. And there's a technique called selectize.js, and R has some things for doing this. What is it called? Auto-completion. But you know, bringing it into the fold so that you're actually finding things that are of interest to you based on some little string that you happen to type. I don't know how to do it other than with selectize.js. Okay? So this is what this package is for, search. Selectize-based search engine for corpora of modest size. What's a corpora? Corpora is what text mining people use to describe collections of text, any kind of collection. In this case, it's a very structured collection in the sense that there's a table. I'm going to refer to that as a document in a corpus. And then we have data structures, S4 structures called doc sets that collect these things together. And here's an example called docset cancer 68. I built this up in a very tentative way. I didn't know whether this would work. So I started out with about 10,000 records. And it works fine. So there's 68 documents for 68 different studies. They have titles. They have accession numbers in the SRA. And then I make this object. And then I can use something called retrieve doc to find the document, all of the fields, for any given study. Okay, these are high-level R things that work using environments and so forth. So you can start to learn about the documentation of the data that's in the sequence read archive. For example, how many variables are used to characterize the different samples? And I was kind of shocked that in this group, some of the data sets are characterized with as few as 4 variables. So it would be an ID, sample ID, study ID, accession number, very few, very little data. And some of them have as many as 25. That doesn't seem like a lot either. And then the types of names that people use, here you go, cell type, cell category, very non-standard. Well, I can't solve that problem, but I can give you access to that information, then you can start solving it, right? Here is what we use, search docs as a function that can take something you want to search for, take a document set, and give you back all the documents that make use of that token. Okay? And so that's a prologue to this Human Transcriptome Compendium. It's a new package in Bioconductor. And it has a vignette. So you can go and start reading the vignette and see how this thing helps you to get access to 181,000 transcriptomic studies. And the cancer is a subset of that. The cancer 43K, or whatever I showed you a few minutes ago, is just a subset of this. And this is in a delayed matrix. This is living in the cloud. And the idea is that we use the summarized experiment to specify selections. For example, I just want a certain number of samples. I know their accession numbers. Then I would pull the columns associated with those. I want certain genes. I can do that all through the summarized experiment operations. So you just use a bracket, ask for the genes you want, ask for the samples you want, and you're done. So that idea is what you can explore using this Google Collaboratory notebook. How many people use IPython notebooks? Yeah. They're nice. Okay? You can use them with Python. You can use them with R. You can probably use them with Julia. And this is a globally accessible one. So I think you have to have a Google identity. But once you get that URL in there, you can start piping your commands into there and get all kinds of wonderful computations done. All right? So I think that's it. No more slides. And it's been a long time. It's been an hour and 10 minutes. So thank you very much. I'm happy to illustrate some of these things interactively to wrap it up. Or you can do them yourself, because everything I've told you in here, you can try out.
Thank you.
Thank you. Questions, comments? I think I'll fire up this 43K just so you see what I'm talking about here. So you can search your studies for some token. Let's say prostate. All right, I'll look up that one. And then it goes and finds all the different studies. Wow, there's a lot of them. Okay, and then you can see the titles here. And then you can click on one if you're interested in it. And then it goes into your cart right there. And you can then download the summarized experiment corresponding to the elements of your cart. And you can load that into R. And the data will not be there. The metadata will be. And then once you've gone and brought that in, when you ask for some genes, it will go out to the HDF scalable data service and get the genes that you're interested in. Okay? So check it out. Break it. Put mileage on it if you're interested. Yes. Should I do my Camtasia now? See if I filled my disk? I don't really, yeah, I think you have to click there to stop recording, right?
Yes.
Okay. Stop recording.